Hey guys, so in our last video, we were talking about how cells divide. So we said that prokaryotic cells use binary fission and eukaryotic cells use mitosis in order to make a copy of their cell. Um, Eukaryotic cells don't just do mitosis. So cell division is just one small phase of a larger cell cycle. So before we can talk about when or how a cell divides, we first need to talk about how does a cell live its life. I mean, imagine if you were to study a whole culture or a whole species and all you cared about was, well, when exactly do they have offspring? Well, who cares, right? There's all this other stuff that goes on in their lives. What about when they grow up? What about interact in a society? How are their packs put together? All sorts of different facets of that organism aside from just reproduction. So we're going to take a second and we're going to look at the whole cell cycle, and then we will consider how a cell might divide. So this is the cell cycle right here, and it starts right in this turquoise section here. It's called the first growth phase. So it's going to start here, and this phase, if you look on the inside of the circle, we have these little letters here. So we have G1, S, G2, and M. So those stand for growth 1, S, which is the synthesis phase, G2, which is growth 2, and then mitosis, M. And all of the phases besides mitosis M are collectively termed interphase. So G1S and G2, G1S and G2 are all collectively termed the interphase between mitotic divisions. Now, it's important to understand here that one of the reasons why there's so much emphasis put on mitosis for a cell is because it's the only time you can see what's happening inside of the cell. The rest of the time, during G1S and G2, it just kind of looks like nothing's happening. There's not a lot of anything. The cell just looks like a cytoplasm and a nucleus, and that's it. Very, very boring. And so M is the phase where we can see what's happening. And so M is the phase that we study most dynamically. But the cell is actually in interphase for most of its life. So right now we're going to talk about the different phases of interphase. So starting with G1. G1 is when the cell's just going about its daily life. These are when the cell's just making proteins or lipids or carbohydrates. It's doing protein synthesis. It's growing. It's metabolizing. It's just living its best life. That's what G1 is. It's just the cell living, going through its day to day. At some point during the cell's life, it's going to hit a critical point here, and it's going to decide, I'm going to divide. Everything from here out, okay, from the end of G1 out, everything from here out is really a committed step towards cell division. So the first thing that a cell needs to do in order to get ready to divide is it needs to replicate its DNA. So the cell is going to take some time and it's going to make a copy of its DNA. Once the DNA is fully copied, then it's going to bulk up. It's going to have to get twice as big basically, because you're going to have to split it in half. And so the cell's going to try to grow real big, and it's going to double its mitochondria and its Golgi and its smooth ER and its rough ER and all the organelles that it has. It's going to double those up, and it's going to get ready to divide them. And then in mitosis, it will go ahead and divide everything that it doubled in the S phase and G2. So in S and G2, we're really getting ready for division. But in G1, the cell is just going through its normal everyday life. Now, G1 and G2 were originally called gaps, gap 1 and gap 2. And this is when scientists believed or described that the cell was in a gap of activity. It was doing nothing. Years later, of course, we know that this is not when the cell is doing nothing. This is when the cell is doing everything. Everything in the cell's life is happening in this G1 phase, except for division. So S, G2, and M are all about cell division. But G1 is really the one where the cell's just going through its normal everyday life. So in our own preparation to understand the cell cycle, we need to take a look back at DNA. So DNA, as you hopefully recall, is the genetic material of the cell. And the DNA is stored in the nucleus of the eukaryotic cell. That's because, remember, any mutations in DNA cause long-term damage to the organism. So our DNA is double protected. We said it was like in the VIP lounge, right? Like you have to get into the cell, but then you also have to get into the nuclear envelope to have access to the DNA. For the majority of its life, DNA exists loosely as something called chromatin. But when it's time to divide, the cell is going to tightly pack that DNA down into chromosomes. In this place, what we need to talk about is that originally the DNA is loosely spread out, then it gets copied, and then it's going to tightly pack down into chromosomes. And the idea about packing it down just really is kind of similar to what you do when you travel. Right now, if I were to look in most of your rooms, I predict that your clothes are not neatly placed away, KonMari in your drawers, all nice and organized and, set up by color and perfectly put together. I predict that your clothes are all over the place. That's because you're living your life, right? You need access to your clothes. We don't want to pack them away in vacuum seal bags because you need to get in and access them every day. But if you were going to travel, doesn't that sound great? Okay. If you were going to travel, you were going to Hawaii, you would pack your clothes into bags because it would be real dumb to show up at the airport, right? Imagine you show up at the airport and you've got just an armload of your clothes. I'm just bringing these clothes and I'm just going to dump them on the conveyor belt. And can you please get them to Hawaii for me? Because I'd really like to have them when I'm there. No way, right? They're not going to make it. They're going to get lost in transport. If you want to make sure they get from point A to point B, you pack them into a bag, nice and tight, nice and correct. And then the bag can make it. And then when you get there, of course, you get to the hotel, you're going to not keep everything tightly packed and organized in your bag. It's all going to kind of explode out of your bag for a while, right? And you're going to be using it. And by the time it's the day before you're coming home, your clothes are probably all over the place again. But then it's time to travel. So you're going to pack them back down into the bag. This is the same concept of the chromatin versus chromosomes. So chromatin are those open drawers, that big old mess of clothes, and that allows access to them. You can try on that shirt, you can get into that gene and express that protein. You want to make sure that you have them accessible. But when it's time to travel, when we're going to do mitosis and we're going to split this DNA into two separate cells, and each cell has to have a complete set of DNA or it's damaged and we're leading on our way to cancer, well, we really better make sure that we pack everything down nice and tidy into the chromosomes. That's like packing them into your bag. And then we can separate them and put them exactly where we need them to go. And then we can unpack when we get there. Okay, so your cell is just being very prudent, very pragmatic. It wants to make all of the DNA into these little chromosomes so that they're very easily bundled, packaged, and separated so that we make sure that we get a full set in each cell and the cells and the organism stay healthy. During the S phase of the cell cycle, we do DNA replication, and we're just going to be focusing for a moment just on this S or synthesis phase. During the S phase, we take one piece of DNA and we copy it so that we have two. Then we can separate these two pieces of DNA into individual cells and end up with the same amount we had before. So if I have a cell and it has one copy, and then I make a second copy, and then I make two cells, they each have one copy. So that's perfect, okay? So what we're gonna do in the S phase is we need to duplicate our DNA before we can divide it. In order to keep it together, kind of like if I make two copies of the same thing, I might stick them together with a little paper clip to make sure they stay together until I'm ready to separate them all apart. That little paper clip that holds the two identical chromosomes together is called a centromere, and it's right here in blue, and it's like a little physical binder that holds the two chromosomes together. So what happens during this phase is that we take a single chromosome, and chromosomes look like these little eyes. We call them eyes and X's. Okay, so it looks like this little eye, and I'm going to duplicate this eye, and I'm going to put two of them next to each other, and then I'm going to paper clip them together using the centromere. So this individual up here is called a chromosome, and these individuals down here are called chromosomes. But when they are together, held together by the centromere, we call them sister chromatids. Everything in mitosis and meiosis is female because it's all about reproduction and like offspring, and like that's a very female role, right? And so we say mother cells and daughter cells and sister chromatids. So everything is female. So we have sister chromatids here. And so we have the individual chromosome gets duplicated and put together at the centromere into sister chromatids. And then during cell division, during mitosis, they get distributed into the two daughter cells, and we get two chromosomes back into the two daughter cells. So it's called a chromosome before and after division. When it's doubled, we call that a sister chromatid. Just some terminology that's really going to help you when you're understanding, when you're reading. You should be reading your book as you're understanding and as you're reading. So at this point, we have talked about growth, right? G1, that's like what we learned about with the cell in the first place. Protein synthesis, lipid creation, making the little secretory vesicles, and the Golgi doing all of its separation. That's all G1. Then we go through synthesis, which is the phase we just talked about where the DNA gets doubled. We're not really going to get back into it, but in G2, all of the rest of the cell material is doubled. Again, mitochondria, Golgi bodies, smoothie R and roughy R, cytoplasm, it all gets multiplied so that we are ready for the M or mitotic phase. So mitosis is nuclear division. Mitosis separates the nuclei. This is important. It's not cell division. It just does the nucleus. Cytokinesis, which we'll talk about later, cytokinesis divides the rest of the cell. So the M phase is the mitotic nuclear division, and it's broken into 4 separate phases: prophase, metaphase, anaphase, telophase, and we sometimes call them PMAT to remember them. So prophase, metaphase, anaphase, telophase, and then finally, after the nucleus is divided, then the cytoplasm gets divided in cytokinesis. And so in tomorrow's lesson, we will be going through the individual phases, prophase, metaphase, anaphase, telophase, and cytokinesis to try to understand this M section of the cell cycle. Okay, so we've done G1, S, and G2. We've done all 3 sections of interphase, and now we're going to be starting this mitotic phase, and we'll be talking about that tomorrow. So I'll see you then.